Influence of short-term storage conditions, concentration methods, and excipients on extracellular vesicle recovery and function. A presentation from Simon van de Wacker, PhD candidate, Department of Experimental Cardiology. UMC Utrecht. Extracellular vesicles are nano-sized particles and make up an endogenous intercellular communication system to transfer active biomolecules like proteins and RNA. Extracellular vesicles can influence the cellular microenvironment and mediate both physiological and pathological processes. These combined properties make extracellular vesicles an interesting source for a wide variability of clinical applications. In order to successfully use extracellular vesicles as a therapeutic, a scalable source of well-characterized extracellular vesicles is required. However, obtaining high yields using current isolation techniques remains challenging. Optimization of the subsequent isolation procedure while ensuring physical stability could significantly contribute to recover higher extracellular vesicle yields. Therefore, in this study, extracellular vesicle recovery and functionality were evaluated after varying different steps and conditions in the isolation and storage process, including choice of buffers, concentration methods, storage tubes, protective excipients, and storage temperatures. In the top figure, an overview of our initial isolation procedure is shown. Briefly, extracellular vesicles were isolated from conditioned medium of cardiac progenitor cells. After centrifugation and filtering steps, conditioned medium was concentrated with tangential flow filtration before injecting the residue into a high-screen captocore column to purify the extracellular vesicles. After elution, extracellular vesicles were concentrated using spinfilters. Extracellular vesicle morphology was assessed with transmission electron microscopy, showing bilayer enclosed vesicles in a size range of 30 to 200 nanometers. Phosphate saline buffer is the most widely used buffer for extracellular vesicle storage, but buffers containing phosphate are known to show pH shifts with concentration changes, or upon freezing and phosphate precipitates in presence of high concentrations of metal ions. For this reason, we explored the use of HEPES saline buffer as an appropriate alternative for extracellular vesicle isolation. Using HEPES instead of phosphate saline buffer during the isolation procedure resulted in improved extracellular vesicle recovery for particle yield but not for total protein, which was confirmed by imaging flow cytometry and Western blot analysis, revealing higher expression levels of extracellular vesicle markers. Concentrating extracellular vesicle samples during or after a purification step is often essential for subsequent analyses or functional studies. However, it is well recognized that during these concentration steps many extracellular vesicles are lost because of adhesion, aggregation, or destruction. The most widely used method to concentrate extracellular vesicles is filtration using centrifugal spinfilters, which vary in membrane materials, pore sizes, and volumes. 4 different types were compared in this study. 15 ml spinfilters containing membranes with a pore size cutoff of 10 kDa gave the highest particle and protein yields compared to spinfilters with a larger pore size or a smaller volume. Furthermore, higher extracellular vesicle recoveries were seen using spinfilters compared to a PEG-based precipitation approach. The findings were confirmed by Western blot analysis of extracellular vesicle markers. Although it is well recognized that extracellular vesicles may be partially lost during storage, due to nonspecific binding to tubing material, there is little known about the influence of different materials on extracellular vesicle recovery. The most widely used materials are polypropylene tubes and glass tubes, with or without specific coatings to reduce adhesion. Here we studied whether tube materials and coating influenced extracellular vesicle recovery after short-term storage. The results provide evidence that storage in hydrophilic and hydrophobic glass tubes may lead to increased extracellular vesicle loss as compared to the different polypropylene tubes. To protect extracellular vesicles stored in PBS against aggregation and adhesion, the use of excipients tween 20 and albumin on extracellular vesicle recovery was explored. Both tween 20 and BSA are known to cover solid-liquid interfaces, and thereby potentially reduce extracellular vesicle loss because of adhesion. Both tween and BSA excipients were able to protect extracellular vesicles from loss during storage, without impacting the promigratory properties of extracellular vesicles on endothelial cells. Recovery of sufficient yields of extracellular vesicles remains challenging as a result of adhesion, aggregation or destruction/decomposition. For this reason, optimization of each step in the downstream isolation process and storage conditions is pivotal. In the table, a comparison of recovery rates between the conditions that yielded highest and lowest recovery for all study conditions is shown. Altogether, this study provides insights on which steps are important to optimize to recover appropriate extracellular vesicle yields. Optimization of extracellular vesicle isolation, concentration and storage protocols helps to increase extracellular vesicle recovery and thereby, to a lower need for extracellular vesicle production upscaling. Better understanding of protective excipients and buffer formulations on extracellular vesicle function will help extracellular vesicle research to accelerate translation of extracellular vesicle-based therapeutics towards clinical application. Influence of short-term storage conditions, concentration methods and excipients on extracellular vesicle recovery and function.